Hi, this is Dr. Eric Spann from What's Up Doc. I've been away because honestly, I've just not known what to say about the ongoing COVID pandemic since September until now. I've honestly been caught off guard recently by the resistance and the fear of many regarding the new COVID-19 vaccines that have just been approved on December the 11th by the FDA. I have found about 15 common concerns or questions from patients, family, healthcare workers, and you see these on the left regarding these recently approved mRNA COVID-19 vaccines for the SARS-CoV-2 virus. Now, these vaccines have been developed in an accelerated program, and you see that most of the common concerns from the public at large have to do with safety, side effects, and honestly some true misunderstandings about the danger and effectiveness of COVID-19 disease from the SARS-CoV-2 virus. That is, versus the infection versus the vaccination. So I've taken in hand to produce 3 brief videos to try to answer each of these and to help answer the other different questions about these new vaccines, including in part the basic science of vaccination, the science of mRNA vaccine development, and the ethics of this process. In part 2, we will address the effectiveness, safety, and the side effects. And in part 3, we will address the availability and rollout of the Pfizer and Moderna vaccines, lastly trying to set the record straight regarding some concerns and even conspiracies spawned by social media, and some common sense closing thoughts, I hope. So let's get started. With those things in mind, let us look at some basic science, and let's get that under our belts first. First of all, immunity to nearly all of the infections for which vaccinations have been developed work through antibodies. Antibodies that are produced by natural infections or vaccination are the basis for lab tests for examples of all types of diseases such as hepatitis, Lyme disease, mononucleosis. When you take a gamma globulin or an IgG shot, that's what these are composed of, are antibodies. Another example that some of you ladies might be familiar with is the RhoGAM injection that a mother who is Rh negative gets when she's pregnant or after she's delivered to prevent subsequent babies from having hemolytic disease of the newborn. Antibodies are the targeted ammunition of immune warfare. You see on the right-hand side of the slide the process of immune antibodies attaching to viruses or bacteria or cells in our body that are infected with either of those, and beginning this chain reaction that leads to white blood cells destroying the infected cells, viruses, or bacteria. So now that you understand a little bit about immunity and antibodies, what exactly is a vaccine? Well, it's really just a chemical mixture of proteins or chemicals that are closely associated called nucleic acids. These are DNA-like molecules that once extracted and purified from viruses, bacteria, or other germs help protect us against future infection or their toxic byproducts. Think, for example, about tetanus and diphtheria. The vaccinations we get for them are actually against their toxins. So vaccines work like this, and you'll see here. Our cells, usually a muscle cell in the shoulder or hip, absorb the injected vaccine protein mixture, then present them to our white blood cells, which cause numerous cell lines of our immune system to go into action. One group produces antibodies, these are the B cell lines generally, and the other group, generally T cells and some natural killer cells, train and prepare the memory cells for future kinds of exposures. Then the next time that they're exposed to the offending organisms or their toxins, these B cells or T cells go into action. The B cells produce protective antibodies that attach and segregate the invaders with a coat of these antibodies for presentation to our killer cells for destruction. Or you see that the memory killer cells are chemically signaled to the area of inflammation and infection to directly attack these pathogens. We are then immune. Sometimes boosters are needed to keep the antibody memory B cells primed or to provoke the memory of the memory T killer cells or the T helper cells. So then how was this COVID-19 SARS-CoV-2 virus vaccine developed? First, realize that these first 2 vaccines are just one of many types of vaccines to come, and we're likely to have 5 or more types of vaccines for COVID-19 or future related coronavirus infections. Some of these you see here are DNA RNA viruses, which give our cells direct instructions for how to target specific viral components. These two vaccines are what we call mRNA or messenger RNA vaccines. For MMR and chickenpox, we use what we call live attenuated vaccines, which means that the viruses are weakened chemically or physically with temperature before inoculation so that they can't reproduce. They only present the viral particles to our cells for the immune process to begin. These inactivated viruses work much the same way, and our polio vaccine is this type. Subunit vaccines, which have been synthesized and purified with just viral component parts, such as for pertussis toxin, hepatitis B, human papillomavirus, and most famously influenza, consist of subunits of the virus which have no ability to reproduce because they have no genetic information, but they allow our immune system to identify and target viruses or bacteria if exposed in the future. Lastly, viral vector vaccines, which are very new, are in development for diseases such as Ebola. These use harmless viruses to deliver segments of potentially deadly viral genomes into our system to present them for sensitization by our immune system. One of the vaccines being tested against COVID-19 is actually a monkey adenovirus. These are cold viruses in humans and in monkeys as well. But this is a monkey adenovirus which has been induced to attach the SARS-CoV-2 viral genes so that we can become exposed to some of these viral proteins without producing all of the molecular machinery with genetic products necessary to allow viral assembly and reproduction in our cells. But let's focus on how we developed an effective and safe COVID mRNA vaccine so quickly. You see here on January the 11th, 2020, the Chinese scientist in the viral lab in Wuhan, China, posted the genetic data for the SARS-CoV-2 virus to the CIDRAP website. Note at the bottom that even in this posting, they very famously falsely claimed that there was no human-to-human transmission, and we sadly lost over a month of warning, which could have presented so much harm, we believe. So if we look at the virus that causes COVID-19 disease, this is a 3D model of the virus with its actual structural proteins, its genetics, its lipid or fat molecule envelope, its membrane, and its RNA genetic information stored inside, and then this spike protein that so many have heard of on the outside that allows it to attach to and invade our cells. As a matter of fact, you see that if you look at this in cross-section, it looks like a crown, and that is why it's called a coronavirus, because the Latin word for crown is corona. So this is the 3D structure of the entire spike protein and the chemical structure of the area at which the SARS-CoV-2 spike protein binds to our ACE2 receptor. Now, interestingly, and some of you may also have heard this is the receptor that blood pressure medications use to attach and relax our chemical messengers from our kidney to our blood vessels, and medicines such as valsartan, losartan, olmesartan bind to this receptor to lower blood pressure. This, thankfully, appears to have not had any deleterious effects on the severity of COVID disease in patients on these medications, or the effectiveness of these angiotensin receptor binder blood pressure medications in patients who were infected with COVID. This was a concern very early, and it was identified and researched so that we realized that it was actually the illnesses underneath the risk factors rather than the medication or this gene receptor or protein receptor on our system. So very quickly you see here that this ORF2 spike protein gene sequence was uploaded and shared worldwide, and vaccine research began immediately in January. And this is actually how that protein sequence looks when scientists work with it in the lab. Now, this is one of my favorite videos. I've been showing this for 10 to 12 years now. This little video from the DNA Learning Center will show how an mRNA molecule, like those we've described above, are sent out of the cell nucleus into the substance of the cell, kind of like the liquid part and where all of the activity takes place outside of the nucleus. We call that the cytoplasm. And in the cytoplasm, how these little messenger mRNA chemicals cause cellular proteins with our cell's machinery to be produced. So almost like a ticker tape, you see that this mRNA molecule goes through the ribosome, it's called, in our cells outside of the nucleus. And then it produces proteins that work like minuscule molecular machines after they adopt their 3D form as they're taken out of the ribosome. Now, of the four vaccines that are ready or nearly ready to come to market, here are our two mRNA vaccines in the middle. The difference is that they differ slightly in the size of the mRNA sequence that's being used to produce a different version of the spike protein. You see there's slightly different designations on the right, one ending in 1, the other ending in 2. So then what's really the difference between the Pfizer mRNA vaccine and the Moderna vaccine that were just approved here on December the 11th for Pfizer, and the one that we believe will be approved in just the next couple of weeks for Moderna? Well, you see those numbers BNT162B1 and the same designation with the number 2 at the end. So both companies began to work on the entire mRNA segment, producing the whole spike protein, these clones of that spike protein really, at the first, and this was called the BNT162B1. But you'll notice that Pfizer also was working on the 16B2 component that produced part of the spike protein, and they chose to focus their research on the section that exactly binds to our ACE2 receptors rather than to produce the whole spike protein. These areas of those proteins are called binding regions. So this binding region is named BNT162B2. The immune effect appears to be nearly identical, but there may be slight differences noted later. No major clinically significant differences in the two immune responses or side effects have been noted so far, though. So you might be curious how mRNA vaccines work. This diagram from Bloomberg News shows the entire process described above, with the spike protein's mRNA sequence decoded, then reproducing and packaging it in special nanoparticles of fat-like material we call liposomes. And these are truly nanoparticles. They're smaller than a micron. These are packaged and stored properly for administration later. Then our cells absorb them. The mRNA is used in our cell cytoplasm to produce and present that spike protein or its ACE2 binding region, the binding region of that protein, to our immune system to produce those protective antibodies. Now, you've heard me say mRNA repeatedly. You might wonder what RNA is. Well, just realize that it's a single strand of nucleic acids made in nearly the same structure and from the same basis as DNA. DNA, as most of you know, is a double helix with base pairs, whereas RNA is a single helix with nucleotide bases that are not paired. DNA produces RNA, and then RNA, at least messenger RNA, is sent out of the nucleus into the cell to be used to encode and transcribe proteins, as you've just seen in that short video. The messenger RNA, because it is sent out into the cell as a messenger, is used not within the nucleus of the cell with no transformation of any nuclear products, whereas other RNA molecules function within the nucleus. And this is going to be very important to some of you before we're done. See, science can be fun. So drawing this part one to a close, see how the mRNA is delivered into our cell as a nanoparticle lipid envelope, then the mRNA is transcribed by our cellular proteins to produce and present this harmless isolated spike protein or component on the cell surface. Noticing that the mRNA never goes into the nucleus. The products of this vaccine do not enter the nucleus of the cell. But do you know what vaccine products do enter the cell? Every flu vaccine and almost all vaccines that you or your children have ever taken. This will later help us undercut one of the major social media false narratives popular recently. Also note that this is not the first time that mRNA vaccines have been created. We've been using mRNA vaccines against cancer cells for years. The technology has been out there since 2004, since the SARS and the MERS epidemic to follow. We've just never used them against an infectious contagion before. Now, regarding the ethics and moral use of these vaccines, I'll share the following from the ethical branch of the Catholic Church presented to the Vatican through the Charlotte Lozier Institute, and a separate report from a large hospital system in North Dakota that gleaned the same information after research. Now, I found this after not a whole lot of searching, but the statement that is most important is the following. The mRNA COVID-19 vaccines produced by Pfizer and Moderna do not require the use of any fetal cell cultures or lines in order to manufacture or produce the vaccine. This honestly, folks, answers the most common concern that I've heard from many of my patients of faith that would prohibit them from taking the administration of a vaccine that used a growth media from cells from aborted fetuses. So take assurance that these are ethically produced vaccines. You now understand how vaccines are produced, what antibodies do, how this vaccine particularly is produced, and our body uses it to produce immunity. And then when we pick up with part two, we're going to discuss this accelerated development program called Warp Speed for all the various COVID-19 vaccines, as well as the effectiveness and the safety of mRNA vaccines that have been approved by the FDA. Thank you so much for your attention. I'll see you soon with part two.